Hi, my name is Brandon Shah, and I'm from McGill University. I would like to thank the conference organizers for selecting this work for presentation. The title of my presentation is Estimating Dispensable Content in the Human Interactome. This work is joint work with Mohammed Gadi. There has been tremendous progress in network biology in recent years. A lot of the efforts have gone into mapping many different types of biological networks as accurately and completely as possible, and these efforts have been very successful. At the same time, biological network data are challenging to analyze because they're very complex. Here I'm showing a map of the human protein interactome generated by Mark Waddell and colleagues, where each node is a human protein and each edge is a physical interaction between two proteins. A question that naturally comes into mind is, why is the human interactome so complicated? In thinking about how to answer this question, it is helpful to draw an analogy between interactome networks and other types of complex systems, such as the computer system. A few years ago, the Apple Computer Company made this commercial, which nicely illustrates two very different types of complexity, at least according to Apple's opinion. To the left, we have Windows PC, and to the right, we have the Mac computer and the associated software. Both of these computer systems are very complicated, but the point is that the complexity of the Mac computer is necessary and justified for its functionality, whereas the Windows PC computer is unnecessarily complicated and bloated. So the question is, which computer system does the human interactome resemble? Does the human interactome resemble the Mac computer? Or perhaps a better analogy would be the Swiss watch, where all pieces are useful, and the complexity of the system is necessary to carry out the function. This is called the adaptive origin of complexity. Or does the human interactome resemble the Windows operating system, which is unnecessarily complex and bloated? By the way, we actually know why software systems are bloated. And this is because it is very easy for software engineers to keep adding new code to the software, and there is very little penalty for selling bloated software systems. In other words, it is easy to grow and much harder to shrink back. This is called the non-adaptive origin of complexity. This distinction between the two types of complexity for biological networks was first articulated by Michael Lynch, Steve Michnik, and colleagues. Now, the key difference between Windows PC and the Mac computer or Swiss watch is the amount of useless junk in the system. So we need to know how much junk is in the human interactome. The word junk was used by Sidney Brenner to describe useless DNA parts that are nonetheless kept in our genome. But the same concept can be extended to interactome networks as well, where junk interactions describe useless interactions that are nonetheless real interactions existing in our interactome. How do we know if an interaction is useless? Well, if we delete the interaction from the interactome, and the cell and the organism do not feel any pain at all, then we can consider the interaction to be useless. I understand that many people may not like the negative connotation associated with the term junk interactions, so from now on I will refer to these interactions as completely dispensable interactions. These two terms mean exactly the same thing. It is important to note that dispensable interactions have a very precise definition. Dispensable interactions are not experimental false positives or errors. Rather, they are real interactions that do exist in the interactome. The hallmark of completely dispensable interactions is that they are neutral upon disruption. In other words, the fitness of the cell and the organism does not change when you delete the interaction from the interactome. Dispensable interactions are different from non-specific interactions or stochastic interactions. Completely dispensable interactions may even carry out real biochemical functions, but we can consider them as non-functional from an evolutionary perspective. Why should we care about dispensable interactions? Well, dispensable interactions do exist, and they're actually inevitable, according to the theory of molecular evolution and population genetics. Moreover, dispensable interactions pose significant challenges for systems biology efforts to understand human disease. So we need to know how many dispensable interactions are out there in the interactome. How do we find out what fraction of the human interactome is dispensable? Well, we have to start from a high-quality human interactome. We then need to precisely delete one interaction at a time, carefully measure the fitness change of the cell and the organism as a result of the deletion of the interaction, which is highly non-trivial. Classify the interaction as either dispensable or not dispensable according to the fitness measurement, and then repeat this entire process many times for every interaction in the interactome. Now, nobody has done such an experiment, and it is unlikely that this experiment will be done in the near future. So, we need to find some other way to estimate dispensable content in the human interactome. The approach we took is based on structural systems biology. The idea is to take the high-quality human interactome network already validated by experiments, then try to build accurate three-dimensional structural models for a subset of these protein-protein interactions using the technique called homologous modeling. In other words, we're not predicting new interactions. Rather, we're annotating known interactions with three-dimensional structural details. Here's how homologous modeling works. We start with a pair of proteins that are already known to interact from experiment. To build a three-dimensional structural model for this query protein pair, we perform a sequence similarity search of the query protein pair against a library of protein complexes with experimentally determined three-dimensional structures. If we're lucky, we will find a protein complex in the library whose sequences are similar to the sequences of the query protein pair. In that case, we can borrow the known three-dimensional structure of that homologous protein complex in the library and use it as a template to build a homologous model for the query protein pair. Here's our pipeline to construct the human structural interactome. We started with two high-quality, experimentally determined human reference interactomes. The Y2H interactome, which consists of interactions identified in Is2 hybrid or Y2H screens that are additionally validated by orthogonal assays, as well as the INT-ACT interactome, which consists of interactions identified by at least two independent experiments as reported in the INT-ACT database. From each of these two reference interactomes, we then build high-quality homology models for a subset of the reference interactions to construct the structural interactome. Next, we use the structural interactome to predict the precise patterns of edge perturbation caused by different mutations, also known as Agile types. Here's an example. The wild-type protein interacts with three other proteins via three different binding interfaces. Here are three different mutations on the same protein, but with different Agile types. The first mutation is located at the binding interface mediating the first interaction, or edge. And as a result, the mutation specifically disrupts the first edge, leaving the other two edges intact. This mutation is called an agetic mutation. The second mutation is located elsewhere on the surface of the protein and is not involved in any binding interface. And as a result, it does not disrupt any interaction at all. This mutation is called a quasi-wild-type mutation. The third mutation is located in the core of the protein and disrupts the overall stability of the protein. And as a result, the entire protein falls apart, and all interactions are disrupted. This mutation is called a quasi-null mutation. Both quasi-wild type and quasi-null mutations are considered non-agetic. These mutation agile types can be accurately predicted by three-dimensional structure-based methods such as fold-X and bind-prof-X. Here is our pipeline to predict mutation agile types. We started with the human structural interactomes. Next, we mapped different kinds of mutations onto the structural interactomes. These include common missense mutations not associated with any disease from the dbSNP database, as well as Mendelian disease-causing missense mutations from the CleanVar database. Next, we used the structural interaction information to predict the agile type for each mutation. We predict that a mutation agetically disrupts a specific interaction if the mutation occurs on the interface mediating that specific interaction with significant change in binding free energy. We then repeated this agile type calculation for all mutations. Here are the results. It turns out that only a small fraction of common non-disease mutations are agetic, that is, disrupting specific interaction edges. In contrast, a much larger fraction of Mendelian disease-causing mutations are agetic. This is true for both Y2H and Interact structural interactomes, and they both agree with mutation agile typing experiments by Mark Bedell and colleagues. This is great, but how do we go from mutation agile types to dispensable content? Well, we can safely assume that common non-disease mutations are neutral and that Mendelian disease-causing mutations are deleterious. In fact, disease mutations are probably mildly deleterious on average because there are also embryonically lethal mutations which are strongly detrimental, but they rarely show up clinically as disease mutations. If we make these assumptions, then the pie charts shown here tell us the probability of neutral mutations being agetic, that is P of E given N. But what we really want is the dispensable content in the interactome, that is, the probability of an edge being neutral, or the probability of an agetic mutation being neutral, P of N given E. So, we need to invert the two sides of the conditional probability. How do we do that? Well, it turns out that we can use the Bayes rule to invert the two sides of the conditional probability. We already have the likelihood term, P of E given N, from the pie charts in the previous slide. We need the P of N term, which is the prior probability for a mutation to be neutral, but we can get that from the literature. There's also the P of E term in the denominator, but it is just a normalizing constant which we can easily get rid of. So we have all we need to calculate P of N given E. Here are more details on how we apply the Bayes rule. We obtain from the literature the prior probabilities for new missense mutations to be effectively neutral, mildly deleterious, or strongly detrimental. Next, we added the pie charts from the previous slide, which give us the probabilities for effectively neutral and mildly deleterious mutations to be agetic. Now, we're missing a third pie chart, which is the probability for strongly detrimental mutations to be agetic, but we can make some educated guesses. Now that we have everything, we use the Bayes rule to calculate the probability of agetic mutations to be neutral, P of N given E. This then gives us the dispensable content in the human interactome. Using this approach, we estimate that about 12% of the Y2H structural interactome is completely dispensable. and about 16% of the interact structural interactome is completely dispensable. We also repeated the calculation using experimental mutation agile typing data by Mark Vidal and colleagues, and found that about 7% of the human interactions are completely dispensable. These three estimates are all consistent with one another. After taking into account the confidence intervals, we can conclude that less than 20% of the human interactome is completely dispensable. To summarize, I hope to have convinced you that dispensable interactions are important to think about. Dispensable interactions are introduced into the interactome through non-adaptive processes such as mutation and genetic drift. We use the structural systems biology approach to estimate that less than 20% of the human interactome is completely dispensable. Our results suggest that the majority of the human interactome is under strong purifying selection. Finally, there is likely a high degree of variability in dispensable content among different interactome subnetworks, so our estimate represents a grand average. I would like to thank Mohammed Gadi for great collaboration on this project. We have a poster on this work as well, so you're welcome to drop by our poster for more details. Thank you very much for your attention.